welcome to this lesson for Advanced Higher Biology, which today is looking at measuring and recording animal behaviour. Prior to now, we've looked at how you can monitor the population of a particular species, and what we're going to be looking at today is how we measure and record those species' behaviours. Before we move on to our next key area, which is all about evolution, and we'll start with a little review of that. So today's lesson we're looking to understand why and how to record animal behaviour. So we're looking for you to be able to list some of the methods used to quantify animal behaviour. Describe what's meant by an ethogram and how it's used. Describe what is meant by time sampling. Explain why anthropomorphism should be avoided in these studies. Now, when we study animal behaviour in school settings, we tend to be restricted to the sort of invertebrate organisms, the woodlice experiments, and these tend to give us fairly predictable responses to particular stimuli when we're doing them under lab conditions. We might also try and quantify certain aspects of human behaviour, so you might have done some memory tests on your fellow pupils. And these are good experiments, they're worth doing. However, in the field, behaviour is a more complex adaptation. And when we look at the behaviour of different species, for example, you can see there's a huge variability in the types of responses that you might see when we are studying different species. Consequently, we need to make sure that we design these experiments very carefully. It's necessary for scientists to study these organisms to begin with, look at the types of behaviour they are seeing, and try to break down these behaviours into very defined categories, and that allows us to produce quantifiable or countable data. Now, these types of studies require data that have been gathered obviously in a factual and an objective manner, and we have three measurements which we commonly use to quantify animal behaviour. The first is latency, and that's the interval of time between a stimulus occurring and the behavioural response. So, for example, the amount of time between the appearance of a female stickleback and the male doing his courtship dance. We also have frequency, and that's the number of times a behaviour might actually occur within the period of observation. So the number of zigzags, for example, that the male stickleback performs in his courtship dance. And finally, we have duration. That's how long the behaviour actually lasts for. So this may not be the entire period of observation, or it may last longer than your period of observation. So how long that male stickleback continues to perform that dance. And when we have looked at these specific types of data, we are recording them in something called an ethogram. So those of you who did higher biology rather than higher human biology would have come across these before. And an ethogram lists really a species-specific behaviour to be observed and to be recorded in the study. So if we are looking at a hamster in a cage, for example, we might have a list of particular behaviours. Now, the issue with this list is it needs to be more clear and it needs to be more defined. So we need to get very specific and very quantifiable about the types of behaviours that we might see. So to improve this ethogram, we would change our list slightly. So rather than sniffs food, we now have the nose is within 2 centimetres of and pointing towards the food. Looking around the cage becomes both paws are up on the side of the cage and the head is moving. Enters the shelter, all 4 feet must be under the roof of the shelter. Climbs on an object, all 4 feet must be on the object. Self-grooms, licks, rubs or scratches any part of own body. Eats, chews on food in any way. So as you can see, that's made our behaviours a bit more specific and much easier to identify. So while the list on the left appears to be fairly straightforward, as you're watching your organism, you might discover that you need to be more specific about exactly what is meant. And again, remembering that any of these studies would ideally be repeatable. You would want some independent replication at some point, and you need to be very clear on the behaviours that you saw the first time to look again for the same behaviours the second time. Now, when we record the duration of each of these behaviours in an ethogram, together with the amount of time we've observed our organism, that allows the proportion of time spent on each behaviour to be calculated in what we would refer to as a time budget. So having studied your organism, you're looking to see how long in the total period of observation did it spend eating, sleeping, moving or playing, and you can allocate a time budget to that. And that allows our researchers to look at, for example, the difference between wild animals and animals in captivity, for example, or looking for any particular changes in behaviour that might tell us about how we need to manage the safety of those animals. Finally, we need to consider the risk of anthropomorphism, and this is when we attribute ideas of human motivation, traits, emotions or intentions to non-human entities. And this can be a problem because different species have different responses compared to human responses. So looking at this example on the left, for example, we've got a smiling happy chimp and a smiling happy person. However, while that's a happy grin for a human, that's actually a fear grin for a chimpanzee. So we need to be quite cautious about attributing our natural capability or desire to put human emotions onto animal faces. Similarly, on the right hand side, we've got a sad face, but in our chimpanzee that's actually a play face. So you can see again it's a very different kind of expression with a very different intention behind it. And consequently, we need to be careful about anthropomorphism. We need to be just very objective about recording the behaviours that we see and not attributing any sort of intention onto those behaviours. So from that, you should be able to explain the methods or list the methods we use to quantify animal behaviour, describe what we mean by an ethogram and how it's used, explain what we mean by time sampling, and explain why anthropomorphism should be avoided in these studies. As ever, feel free to get in touch if you have any questions, either by leaving a comment, visiting the website, or sending me an email on the address to follow.